So, do you hear me, everybody? The microphone is not working.
No, it is.
It should work.
Yeah, we got the thumbs up. They hear us.
So, just a little test. If you hear, I mean, it feels like you have microphone sound. Yes. Okay. Yeah. So I think now we have to. I'm sorry for disturbing your nice conversation here. Okay. Where is the? Have you seen the opponent? No. Yeah. Yeah. We will have just 10 seconds of a break here. Great. No, you are. So, welcome everybody today. First, I will actually speak Swedish. This is the procedure. We'll switch back soon. Så välkomna till Marija Dubackic disputation i ämnet fysikalisk kemi. Mitt namn är Karin Schelén och jag är från avdelningen för fysikalisk kemi. Mitt namn står här och även e-mailadress. Jag kommer vara ordförande här idag. And now, nu går jag tillbaka till engelska. Now I'll switch back to English. So my name is Karin Schelén. I will be the chair of the today's defense, where Marija Dubackic will defend her thesis entitled Alpha-Synuclein, Amyloid Fibrils and Interactions with Lipid Membranes. My name is here and the email address. In case you have technical problems, please write this email and ask, and I will help you. So we will now turn to present the important persons except of Marija. The next person, which is we are very happy to introduce, is the faculty opponent. And he is present on Zoom, and it's Professor Dr. Raffaele Metzenga from Institute of Food, Nutrition and Health, Department of Health Sciences and Technology at ETH Zurich in Switzerland. And he is also the head of the Laboratory of Food and Soft Materials. And it's a true pleasure to welcome you to this event, to Lund via Zoom. And I would like to thank you for taking your time to read Marija's thesis and prepare for the discussion that later will take place. We have also members of the examination committee, and they are present on Zoom, either on Zoom or in the hall. First, we have Associate Professor Vito Fodera from Department of Pharmacy and the Copenhagen University in Denmark. Welcome. The cameras are a little bit, so welcome.
Thanks.
Then present in the hall, we have Professor Susanna Horsfield from Division of Biochemistry and Structural Biology and the Department of Chemistry here in Lund. Welcome. And finally, Associate Professor Anela Seddon from Bristol Bristol Center for Functional Nanomaterials at H.H. Wills Physics Laboratory, University of Bristol in UK on Zoom. Welcome also. We have also deputy member of the examination committee here in the hall, Professor Marija Dubackic. She's from the Division of Theoretical Chemistry, Department of Chemistry here in Lund. Welcome. I would also like to present Marija's supervisors, Professor Ulf Olsson, who is sitting in the hall from Physical Chemistry Division, Sara Linse from the Division of Biochemistry and Structural Biology, also sitting in the hall. And then we also have Emma Spar from Division of Physical Chemistry, also sitting here. We are happy that we are all together. So today's event, the procedure of today's event is that I will first give the word to Marija, and she will have chance to make the last minute corrections of her thesis. Then I will give the word to Raffaele, who will present a short overview of the research field of Marija's thesis, and thereafter to Marija, who will summarize her work and her main results. We will then have a short break, 5-10 minutes. We will also organize a little bit some technical matters here, and you will also stretch your legs. Thereafter, Raffaele will lead a discussion with Marija concerning her thesis and ask questions. When they have finished, the word will be given to the members of the committee that will ask complementary questions. And thereafter, you in the Zoom or in the hall, the audience will be able to ask questions. Concerning the Zoom questions, please, I will instruct you on how to do later. You can use your hands there. After this, I will close the open part of the event, and the examination committee will withdraw. We have a special Zoom link for that. We will also take that later. And the rest of you will be welcome up to the coffee room who are here, to the coffee room of the physical chemistry for some snacks and refreshments. So then I would like now to give the word to Marija and ask if you have any changes or if you want to redraw the thesis and end this session.
Okay. No, I will try to defend the thesis, and I have some corrections that I would like to ask you to spread. Here you go. Thank you.
And this will be distributed here in the hall. And during this, I think now, we will soon, maybe if I see Raffaele, yes, there you are.
Okay.
We will organize now a little bit while the others are getting some small changes, corrections. We are pinning you so that you will be seen on the main screen. Just a moment. Just a moment. And thereafter you will be able to share your screen. So can let's see. We
terminated for good.
Yeah. Okay. So yes. Okay. So Raffaele,
yes.
Do you hear me?
Yes, I do. Do you hear me?
We don't hear Raffaele.
No.
Just a moment. We don't hear you yet. No. Let's see.
Strange.
We will see how to do.
I think my microphone is working.
No, we don't hear you. We don't hear you. We will just a moment.
Raffaele, it's working. Your microphone. I can hear you on Zoom.
Yeah.
No?
Can you please say something, Raffaele?
Yes. Can you hear me?
No.
I think the problem.
Okay, we don't hear you.
Zoom.
So we will see what to do. This was interesting.
Yes, we're not checking for that.
Yeah, there's an excellent one.
Yeah.
Oh my God.
Okay. So
Now?
yes,
Raffaele?
Raffaele?
Can you hear me now?
No.
No.
Okay.
He wrote them.
Yeah, it works on. I mean, we see your message. It's the problem in Zoom. Yes, the micro. Yes, I mean everything here looks. I mean, okay, just a moment.
And now?
Please say something, Raffaele.
Good afternoon. Can you hear me? I see my microphone is working.
Okay.
Hello.
And is there like,
Yeah. No, this
No,
one.
it's muted. Yeah.
No.
it's up, it's up.
Yeah, now it's up. But this is.
Hi, Raffaele. Maybe we should have a
Hi,
small chat in
Anna.
the meanwhile. It's
Yeah.
so
How
good
are
to
you?
see you.
Okay.
Nice to see you.
Nice to see you.
Yeah, I have turned off my camera.
Now a lot of people are speaking, and I hope now we hear Raffaele.
Good afternoon. Can you hear me now?
We are very happy to hear you.
Very good.
Very
Okay.
happy.
So good afternoon.
Yes, welcome. Please, you may now share your screen,
Okay. Let me
and
see.
I will turn off.
There we
Yeah,
go.
we can just mute ourselves, I think.
Oh no. No,
We do not see. We see presenters view.
We are seeing your presenters.
Okay. I think I can.
Yeah,
fix
that's
it.
good.
Okay,
That's
good.
perfect. Yeah, this is perfect. This is just a
right.
moment. We are setting so we can see you on the side.
Hmm?
Yeah.
Should I mute this computer?
Yeah. Okay. So please, Raffaele, start.
Okay, so good afternoon, everyone. And so I'm very pleased to be in this PhD defense of Maria. I'm very sad not to be in Lund. I was really hoping to be there. I have a lot of friends. I love Lund. Last time I was there, though, we made a quick calculation with Ulf. I came by boat. I had to go through Copenhagen to Malmo with the ferry. So we calculated it was about 20 years ago. So it was for me time to go back to Lund. But unfortunately, with the Omicron thing, I will have to be stuck in quarantine for 10 days coming back. So hopefully next time we will be in person. So I just want to give a quick and brief introduction to the topic of amyloid fibers at the interfaces, just to set the stage to what Maria will discuss more extensively in a few minutes. Just to the crowd which is listening, a couple of words to introduce amyloid fibers in general. So amyloid fibers are a very peculiar and interesting type of aggregate of protein or peptides, which are a hallmark by which you can easily recognize them. They have they have beta strands which are oriented orthogonal to the fiber axis. And this is common to all families of amyloid fibers found. Which give a typical cross beta pattern in the X-ray. So it's very easy to recognize. The beta strands are orthogonal to the fiber axis. The beta sheets are parallel to the fiber axis. So that's a signature on the X-ray that allows to distinguish them very easily from other beta strand aggregate like silk, for example. And there are mostly three families of amyloid fibers: the pathological amyloid fibers, which were the first class of fibers discovered. These are associated with neurodegenerative disease or systemic amyloidosis. And if you go to Wikipedia, you will find more than 20 different amyloid-based disease. More recently, they've been recognized in the context of important functional roles in the cells, and this is the class of functional amyloids. Or amyloids can more commonly be produced in the tube in vitro by starting virtually from any possible protein. And this is the class of artificial amyloid. So as Marija will discuss a lot about the first class of amyloids, just a couple of words about the artificial amyloids. I am in a food department. So we actually do produce amyloid from food protein, food grade protein. Our favorite one is beta-lactoglobulin, which is the most common globular protein found in milk. And you see, this is more or less the shape in the native states. But of course, to turn this protein into amyloids, you have to unfold it. So you start from the native, you unfold it. What we do is typically we use high temperature, and then we hydrolyze. And to do that, you can either do it enzymatically or with the use of pH. And then the small fragments then reassemble into beta strands, as I just mentioned in the beginning, orthogonally oriented over the main fibril axis. They form peptide sheets, and then the sheets sit on top of each other, forming protofilaments. The protofilament aggregate, twist, and form mature amyloid fibril. There are a lot of examples of, say, food grade or artificial amyloids, starting from many different types of globular proteins. Now, the thesis of Marija is more specifically focusing in one of the most infamously notorious protein generating amyloid fibers, which is alpha-synuclein. Alpha-synuclein is a protein which is mostly intrinsically disordered. It has a part which is close to the N-terminus, which is in the alpha helix configuration, but most of it is unfolded and is overexpressed in neurons, and might be overexpressed, and especially is implied in the Parkinson disease. Here I'm showing you a few contrasted stain optical microscopy images that I took from Wikipedia of Lewis bodies, which are involved in alpha-synuclein pathologies, mostly in the Parkinson case. Now, of course, you appreciate that in vivo assembly is significantly more complex than what you would do in the test tube, especially because there is the presence of biological interfaces. Any cell will provide lipidic interfaces which will modulate the assembly of alpha-synuclein in the fibrils. In fact, even if you choose the simplest conditions in the lab, trying to mimic an interface, you can think of a fluid interface like an air-water interface. There are ways, there are tricks to prevent a free interface, free air-water interface, or at least to replace it with a liquid-solid interface. And you see, this is THT emission, which is essentially used to monitor the kinetic of the assembly. You see that without an air-water interface, the aggregation is extremely slow, and as soon as you have an interface, this is catalyzing the aggregation. This is also a secondary or heterogeneous nucleation process, and you see that these are two different cases of THT, and this is a significantly faster aggregation. And of course, if you now pick up the amyloid of alpha-synuclein, which form at the air-water interface, that you can simply do if you approach the liquid-air interface with the parallel substrate, which is called the Langmuir-Scheffer technique, you see you have a lot of amyloid fibers. If you try to go with a needle, pick up something in the solution, you see that there are significantly lower amount of amyloid. So really the interfaces are crucial. And this, the one I'm showing you here, is the simplest interface you can think of. Of course, Maria engaged into a task which is significantly more challenging, because she's trying to see how an interface, which is a bilayer of lipid, that will modulate the assembly. So already from a physical point of view, this is a very complex interface. It's neither a liquid-liquid interface nor a truly solid liquid interface, because the lipids can diffuse. So it's a complex task, and it gets even more complicated if you try to understand more at the say atomistic level how this interface is formed. And to do that, Maria has explored a number of lipids. I think these are 4 of the 6 or even more lipids that she has been looking at. And she's been doing that by combining a very nice and original set of techniques, which go from it's mostly scattering, but also secondary structures, development, and so on. And since I know that Maria will for sure shows a lot of scattering curves, I just want to conclude with one of the first scattering profile that we collected on beta-lactoglobulin amyloid fibers. And you see, this is combining 3 different techniques. This goes back to the time I was with Peter and Anna in Freiburg. And I want to show this slide also to nucleate the discussion, which will come with Maria in a few minutes. But it's just to emphasize that this is a very complex topic that Maria has tried to tackle from different angles. And with that, I'd like to stop here and looking very much forward to the discussion. Thank you.
Raffaele, now it's time for Maria to continue. Maria has her own PC here that is
yes,
handled very well.
Actually, maybe unpin, Raffaele.
Yeah, we unpin.
Where is
You go.
open?
Yeah. Okay.
Yes.
Okay.
No, yeah. No, it's fine. Okay, thank you, Professor Schillen, and thank you for Professor Mezenga for the very good introduction. So now I will try to summarize 4 years of research in 20-minute presentation. My PhD thesis is titled Alpha-Synuclein, Amyloid Fibrils, and Interactions with Lipid Membranes. I would like to start by explaining the title of the thesis. So as Professor Mezenga has already said, amyloid fibrils are protein aggregates, very well-ordered protein aggregates that are, some of them are implicated with a diverse group of diseases. Some of most common diseases are Alzheimer's and Parkinson's disease. And they also possess the important characteristic, and that is that they possess cross-beta structure, which means that they have beta sheets that run parallel to the axis of fibril. Another player in my title is alpha-synuclein, which is an amyloid-forming protein. and it is expressed in brain, and it can in vivo be present as a random coil, or part of the sequence can adapt alpha helical conformation when they are associated with lipid membranes. The role of alpha-synuclein is not fully understood, but lots of evidence suggests that it interacts with lipids and lipid membranes. And lipid membranes are barriers present in all mammalian cells. They are composed of lipids, which are amphiphilic molecules. And that means that they have hydrophilic and hydrophobic part. So when in aqueous environment, which is also in our bodies, they self-assemble into bilayers in order to shield their hydrophobic part from water and expose their hydrophilic part to it. The model membranes I investigated in my thesis are lipid vesicles and lipid disc micelles. Both of them have lipids assembled in these bilayers. And in case you were wondering, why am I investigating these interactions with alpha-synuclein and lipids, is because both of them seems to be implicated with Parkinson's disease. And the Parkinson's disease is the second most common neurodegenerative disease, which is characterized by progressive death of dopaminergic neurons, as well as by the presence of abnormal intraneuronal inclusions called Lewy bodies. And Lewy bodies are spherical aggregates, main component of which are these alpha-synuclein fibrils. And they compose the skeleton of these Lewy bodies, but they also have various lipids have been found to be seemingly trapped in this skeleton of Lewy bodies. So the pathogenesis of Parkinson's disease remains poorly understood. On the higher level, it's supposed to start in the gut or in the nose, but on the molecular level, it's believed to be caused by disruption of membrane during some abnormal alpha-synuclein membrane interactions, possibly during the formation of alpha-synuclein fibrils. And one of the proposed mechanisms of the cell death is that there is extraction of lipid molecules from the membranes, which leads to formation of pores and to the death of the cell. So now that I maybe justified my research, I would like to show you some of the results. And my thesis can be divided into two parts. In the first part, I am investigating in more depth how alpha-synuclein interacts with lipid membranes, both because the biological role, which is not fully understood, involves interaction with lipids, but also because both seems to be implicated in Parkinson's disease. In the second part, I am investigating interaction of fibrils, and that is performed at conditions where fibrils aggregate into the clusters which resemble fibrillar arrangement in Lewy bodies, with the goal to possibly gain some more insight in the formation of Lewy bodies, which is also not completely understood. So in the part one, I investigated interaction with lipid vesicles and lipid disc micelles. Vesicles were composed of oh my God, I don't have my pointer and I'm pointing. So the vesicles are composed of POPC and POPS lipids, and disc micelles are composed of DMPC and DMPS lipids. The difference in between these DM and PO lipids is in the length of the hydrocarbon chain, making the longer PO lipids assemble in vesicles and shorter DM lipids assembled in lipid disc micelles. And in order to mimic the biological membrane, which mostly contains neutral lipids and presence of some small amount of negatively charged lipids, in these model membranes, I had 70% of neutral PC lipids and 30% of negative PS lipids in most cases. And so I also investigated interaction of monomeric alpha-synuclein because it's believed that biological role involves the monomeric alpha-synuclein. And then I also investigated how does fibril formation affect these model membranes, as it's proposed to be one of the mechanisms of Parkinson's disease. So to start with the monomeric alpha-synuclein. As I mentioned in the introduction, alpha-synuclein is a random coil in cytosol, but it adopts alpha helical structure when it is associated with lipid membranes. And there is a convenient experimental technique for investigation of this adsorption, and that is circular diachrist spectroscopy, where spectra of protein in alpha helical conformation significantly differs from spectra of protein in random core conformation. So by analyzing, for example, the spectra at wavelength, a value corresponding to this minimum, which is signature of alpha helical structure in the presence of different lipid concentration, one can learn how much of alpha helices is formed when alpha-synuclein is incubated with lipid membranes. And from this figure, one can draw a couple of conclusions. From this linear dependence on the CD signal on the amount of lipids, and this is lipid to protein ratio, so on the x-axis. So from the linear dependence, we can say that we have same amount of alpha helices per vesicles. From the saturation level, which is lipid to protein ratio, where we have all alpha-synuclein adsorbed to the membranes, and which happens at lower lipid concentration when vesicles contain 50% of charged lipids compared to the vesicles containing 30% of charged lipids, we can say that this adsorption is dependent on electrostatic interactions. And that the value of the CD signal at this plateau is equal in both cases, mean that in both cases we have the same amount of alpha helices formed. We also learned in this project that these vesicles coated with alpha-synuclein are stable structures. However, we introduced formation of alpha-synuclein fibrils by addition of already formed fibrils, which in some cases can act as seeds for new fibrils to form. We see that after the fibril formation has finished, we observe fusion of vesicles, and where we see approximately 3 to 4 vesicles fusing together. And this is a work in progress, so we still don't understand why we see this fusion, how it is happening, and when it is happening. Then we also wanted to compare alpha-synuclein fibrils that were formed in the presence and the absence of lipid vesicles. So we employed X-ray scattering technique. And there is a very lucky coincidence when scattering X-rays from these POPC, POPS vesicles. And that is that the only contribution to the scattering profile is this broad maximum, which corresponds to the bilayer scattering. And which means that this, when we have both vesicles and fibrils in solution, the vesicles won't influence the scattering profile of fibrils, so we can easily see the structure of fibrils. And these vesicles were fitted with the core multi-shell model. Then I'm showing in black the alpha-synuclein fibrils that were formed in the absence of vesicles. That data was successfully fitted with a simple cylinder model. And in the red, I'm showing the fibrils that were formed in the presence of vesicles. And this data was fitted with the superposition of the core shell model and of the cylinder model using the same parameters as in pure vesicle and pure protein case, which means that integrity of both bilayer and fibrils is not affected by this fibril formation. And we conclude that there is no incorporation of lipid molecules into alpha-synuclein fibrils. Here I'm showing the wide angle regime, where one can see the distances in beta strand distances in beta sheets. And you can see that this peak is identical in both cases, which also implies that there was no incorporation of the lipid molecules in alpha-synuclein fibrils, as the distance of beta strands is not affected. Now, I would like to tell you a little bit about how alpha-synuclein interacts with these different model systems, which are lipid disk micelles. And these lipid disk micelles are composed of two different parts. One is the flat and one is curved. And we believe this charge the MPS molecules to be accumulated in the curved part. From the cryo-TEM image, where I just highlighted the disk due to the low contrast, so it's easier to see, we can see that these disks are more or less circular. And then we add alpha-synuclein to these disks, and we see immediately that disks assume rather very elongated shape. We attribute this shape transition to adsorption of alpha-synuclein monomers. And by we can also see that the area of the flat part is decreasing, whereas the area of the curved part is increasing. We can tell that alpha-synuclein adsorbs preferably to this curved part of the discs. We also wanted to follow this fibril formation in the presence of discs in real time. And for that, we employed contrast matching small angle neutron scattering technique, which is very convenient technique to investigate multi-component systems. And it's based on the principle that hydrogen and deuterium interact very differently with neutrons. So by varying ratio of H2O and D2O in the solvent, one can match out one of the components to the solvent. And in our case, we did match out the protein component so that we only observe scattering coming from interaction of neutron and this disc micelle, so that we can follow what's happening to disc micelle when alpha-synuclein fibrils are formed. So here I'm showing time evolution of the scattering profile of lipid disc micelles after they've been incubated with alpha-synuclein. And I'm comparing it to the profile of discs alone, which is the black curve. You can see at the very early points, the scattering profile is significantly different, which is also what we see from cryo-TM, where we have elongated shape. But the scattering profile in time starts to resemble the scattering profile of the pure disk dispersion, which means that the disks start to recover to the initial shape. And we attribute this recovery to the desorption of alpha-synuclein monomers and their incorporation in the fibrils that are being formed. And interestingly, these scattering data obtained at different time points were successfully modeled with a linear combination of scattering profile of pure disks, a scattering profile of disks just after they've been incubated with alpha-synuclein, where you have a very elongated structures. We call this alpha recovery parameter, which tells what is the fraction of disks that have been recovered. And we see that this recovery parameter reaches 0.7, which tells us that after approximately 8 hours, 70% of the disks is recovered, and that value is not changing in the 21-hour experiment. So we conclude here that by modeling this scattering profile with this linear combination, we have presence of both recovered and elongated structure at different time points, which tells that the desorption of alpha-synuclein monomers happens in all or none case. So first, all monomers are desorbed from one disc and it recovers to initial shape, and then from the other, and so on. So now I finished with the part 1, and I would like to tell you a little bit about part 2, where I investigated the interaction of fibrils, between fibrils. And these interactions were performed at conditions where alpha-synuclein fibrils are not colloidally stable, so they aggregate in these dense clusters that are prone to sedimentation. This condition involves pH close to alpha-synuclein isoelectric point, so the protein is not charged enough to be stable. And we wanted to characterize these clusters from this pH of 5.5. And we employed small angle neutron scattering. Here I'm showing scattering from 19 samples prepared at slightly different conditions and measured at 3 scattering facilities. And in all contain these alpha-synuclein clusters. And there is in all the sample, there is only power law regime, where scattering intensity depends on scattering vector in a power law. And this value of the exponent can be considered as the fractal dimension of these clusters. We find this value to be equal to 2.6, which means that we are dealing with mass fractals that are formed due to presence of some attractive interaction between fibrils. And we attribute this interaction to be hydrophobic interaction. Then we also wanted to see if we can reproduce what we see, this fractal dimension, in a simple model where we randomly place fibril of random orientation, and we calculate scattering profile of this of such clusters. And we are able to, and then by analyzing these profiles, we see that we are able to reproduce this fractal dimension to the good approximation. Then finally, as I mentioned, that Lewy bodies are composed of skeleton of alpha-synuclein fibrils and some lipids that are seem to be trapped inside. We wanted to see what is happening to lipid vesicles when these clusters are formed in their presence. So we let fibrils form, and after the fibril formation has finished, we also centrifuge the samples to make sure that everything that sediments will actually sediment. And then we analyze the supernatant, and we look for the amount of lipids in the supernatant to conclude how much has sedimented. And we were able to see the sedimentation occurring in most of the cases. However, we were unable to see any clear trend. So this is as well a work in progress, and we still need additional experiments to make some solid conclusions as to if there are some specific interactions between these membranes and alpha-synuclein fibrils that could explain Lewy body formation, gain some insights in Lewy body formation. And now I would just like to remind you of the main conclusions of the thesis. So when investigating interaction of alpha-synuclein and these vesicles, we see that alpha-synuclein adsorbs and form alpha helical and adopts alpha helical conformation. We see that this adsorption depends on electrostatical interactions. And then we also see that these coated vesicles are stable structure. However, when the fibril formation is induced, we see fusion of vesicles. Then also we, by comparing fibrils formed in the absence and presence of vesicles, we conclude that there is no incorporation of lipids in the alpha-synuclein fibrils. When we investigate fibril formation in the presence of the lipid disc micelles, we see that alpha-synuclein at the beginning adsorbs, which results in the elongated shape transition from circular to elongated disc micelles. But when alpha-synuclein is desorbed, the discs are able to recover back to their initial shape. And this also seems to be all in non-process, which means that first one disc is free of monomers, then the second one, and so on. And in the part 2, we see that colloidally unstable alpha-synuclein fibrils are arranged into mass fractals, which indicates that there is a presence of some attractive interactions, which we think is hydrophobic interactions. Then we also were able to see co-sedimentation of lipid vesicles with alpha-synuclein fibrils in most of the cases investigated. And I would also like to thank to some people, to my supervisors, my closest colleagues and collaborators, to my supervisor at Newton facility at NIST, where I spent 3 months during my first year of PhD, my graduate school, Swedness, and the funding agency that funded the school, and hence my PhD journey. And I would like to thank all of you for your attention.
Thank you, Marija. Thank you very much. So, yeah, I think now we will take a break until, what you say, 10:02? We have time. Okay. So we will arrange, and you wait a little bit. Okay.
Hey.
So, hello, everybody. Yeah. Well, then the next interesting part of the dissertation is now when Raffaele and Marija will start with the discussion. So if everybody is in here in the hall, yeah, just a moment. Okay, very well. Please, Raffaele, you may start.
Okay, so first of all, I'd like to start by congratulating Marija for having done a very commendable work to try to understand how the presence of lipidic interfaces modulate the presence, the aggregation of alpha-synuclein. So I have a number of questions that I would like to ask, and I am referring to the PhD document, to the PhD thesis. So just, okay, it's good that we are on the same, literally on the same page. So the first question I'd like to ask, or better to understand, is how do you explain, I would like to hear again, what is the mechanism by which you have a conversion of micelles from disc-like to elongated? I think the data are clear, both the scattering and the cryo-TEM, but I'd like to understand better what is the mechanism according to you.
So we believe that alpha-synuclein adsorbs on the rims. Because we also believe due to electrostatic repulsion, which would be preferably have charged lipids on the rim.
Yeah.
And we know that alpha-synuclein adsorbs on the charged membrane surface, but yeah, there could also be a little bit maybe of a hydrophobic interactions, as we would have some more exposure. So it's reasonable to assume that it adsorbs on the rims, and then if it preferably adsorbs on the rim, maybe there's a tendency of disc to change the shape so it can accommodate more alpha-synuclein molecules. So it becomes more elongated where the curved area is increasing, and the flat one is decreasing. And also it could be preferable for alpha-synuclein to be adsorbed on these long surfaces, because then it can form a full alpha helix where all first 100 residues can accommodate into extended alpha helix rather than a broken helix, which would be on shorter surfaces.
Yes, this is for sure. I will go to the alpha helix in a second. Yes, of course, I also agree on the fact that this switch of shape is induced by the absorption of the alpha-synuclein. But reading from the thesis, I have the impression you hypothesize that the absorption is on the rim of the.
Yes.
And for me, I would like to understand more what makes you think that they will absorb at the rim. Why will be beneficial to absorb at the rim?
Yeah, but I mentioned before due to electrostatic interactions, if we have more charges on the rim. So I think that is probably the main contribution.
Yeah, I mean, for sure on the electrostatic, but I don't know whether you are familiar with the concept of the OK pack. I mean, there is an absorption of, say, block copolymer on surfaces. Actually, implies that there is an entropy of the random coil that we will agree will be the dangling end, and if you accommodate all the alpha-synuclein at the rims, there will be an overcrowding there. So that you will, I mean, I agree on the electrostatic, but then you pay back with the entropy. So at some point, this will not stand. And you think this is at the moment where you have a triggering from disc-like to elongated, or?
Well, maybe this results in getting elongated structure to some extent. Otherwise, maybe they would elongate even more if we didn't have to pay entropic penalty. But I believe that the recovery, hence the desorption, is more due to alpha-synuclein getting incorporated inside the fibrils, because we have a fibril formation parallel to this.
On the surface of the on the surface of the lipids, you mean? Or?
That can also be, yes.
Mm-hmm.
It's possible.
I mean, I agree with most of the story. I just think that even if the alpha-synuclein will absorb at the surface, you will still see an elongation from disc-like to elongated bilayer, simply because you are bringing charges. Initially, you neutralize charge, okay, but then I mean when an object is overcharged, it's like polyelectrolyte. They elongate, they stretch because of charges. So I do not see a real evidence of alpha-synuclein absorbing at the ridge, and for me it's not needed. It's just the absorption will anyhow trigger an elongation of like in any polyelectrolyte. So I think there's no need to live on this assumption. But yeah, this will make sense anyhow. So it's more subtle is why will the alpha-synuclein absorb at all? So let's talk about electrostatic. What is the so remind me on the paper 1, we're looking now at pages 78, 79. Remind me, the pH is above the isoelectric
Yes,
point, right?
but not too much, yes.
Yeah. So so is the protein will be barely negatively charged.
Yes.
It's still absorbed on a surface, and you show it convincingly. I don't know what this is, or the following chapter you have shown in the presentation. The more charge bilayer will receive higher absorption of the alpha-synuclein. Isn't this surprising to some extent?
Well, I think it has, yeah, if we think of the alpha-synuclein as a whole molecule, but the charge is alpha-synuclein molecule is very polarized, where we have N-terminal region which contains mainly positive charges, and C-terminal region that contains mainly negative charges. So if we consider that this positively charged N-terminal region interacts with the membrane, which are negatively charged, and yes, we think so, because also it is this N-terminal region which adopts alpha helical conformation, whereas C-terminal region stays as a random coil. Yeah. So in that sense, it makes sense that it absorbs a negatively charged vesicles.
Mm-hmm. I mean, anyhow, protein are a strange object. You know, they always have positive, ampholytic. So I mean, they can also absorb if they are overall light charges. I mean, especially if you have a moderate surface charge density on the. So now one thing is, can you remind me, because you have a complex lipid formulation. I recall you have about one alpha-synuclein molecule absorption every 200 lipids, something like that.
Yeah, it's in the other manuscript where we have
1 to
vesicles.
200, yeah. But can
No,
you
it's
tell
1 to
me?
500 in our case.
uh,
Oh, sorry. Well, depends on the charge of the membrane.
Yeah, yes,
Yeah,
that's what
yeah,
I want to
yeah.
know. How much, when you have 1/200, how much charge, how much negatively charged is the lipid surface?
So
Or-
if we're looking at 200, then it would be 100 charge lipids. If we are talking about saturation point at 200, because that happens when we have 50% of charged lipids. And when we have 30% of charged lipids, then it happens around 500. So then we have 30% of 500.
So it's quite charged.
Yeah.
I mean, I have no problem to accept that. I mean, for sure that I think you have other indications of electrostatic absorption. Yeah, I mean, maybe I can actually point to that indirect evidence, which is the formation of the alpha helix on the. So I think this is beautiful. The CD you have is it's truly beautiful. You clearly show that you go from a random coil on swimming in solution to a very nicely organized alpha helix. Do you happen to have, so how do you explain that? First of all, why would you have an alpha helix once you have absorption on the interface?
So there is this specific arrangement of residues in the sequence. And that allows for, so when alpha-synuclein forms alpha helix, on one side we have hydrophobic residues, and that we believe to be incorporated into bilayer, so interact with the tails. And then we also have positive charges on two different sides, and that interacts with the head groups of the bilayer. And then we have negative charge that are sticking into solution. So that becomes very energetically favorable to form alpha helix.
So do you think that charges modulate the formation of alpha helix?
Both, I think it's both charge and hydrophobic interaction.
Well, you know, I mean, polypeptide, if you think, for instance, polylysine or polyglutamic acid, you can have them both as a random coil or as an alpha helix. And this will depend very much on the linear charge density. If you neutralize the charges, you will give to the peptide the possibility to form alpha helix. If the object is heavily charged, alpha helix cannot exist. So I think once you absorb the N-terminal on the surface, you will neutralize the charges and you will have then a possibility to form alpha helix. The indirect proof will be, have you done or have you measured the CD of the alpha-synuclein in solution as a function of pH?
No, that's a good idea. Yes.
Because I mean, it will be an indirect proof if you will be able to show that when that block of that part of the protein is mostly neutral, you will have secondary structure forming.
I mean, yeah, it isn't quite known from the literature that alpha-synuclein won't absorb onto neutral membranes.
Yeah.
So there needs to be some charge in the membranes.
Yeah, I mean, my point is that if you will manage to absorb alpha-synuclein on a neutral charge, you will probably not see alpha helix. Am I right or am I wrong?
Yeah, I mean, now I'm pretty sure that CD were done and that there was no alpha helix signal. So that when we have when they were neutral membranes. And yeah.
Okay. Good. And then the other thing is about still on that chapter, page 97. You give a schematic or highly idealized picture of how the alpha helix will appear on the surface of the bilayer. First question is, do you really trust it? I mean, this is, of course, we agree it's highly idealized, but do you really think this is what is happening? Because you make a parallel to a 2D isotropic nematic transition.
I mean, if it's, I think maybe there should be some more concrete quantification as to how many exactly molecules absorb. But if it's really this many molecules per this area, then I don't see how else would they fit if they wouldn't align.
Well, I mean, you know that I've been working on the isotropic nematic transition in 2D, and it's a very complicated topic. First of all, people are still not agreed whether this is a 2D or 1D order transition, but both in the assumption that you are at the equilibrium. But here you will have something which is a strongly non-equilibrium process, because you have absorbed an helix, alpha helix, and up to here we agree. But this alpha helix, first of all, is highly flexible. Okay, it's still that doesn't have the excluded volume of a fully standard alpha helix. Then when you absorb it, and we agree on electrostatic, you have no possibility to do free rotation of the alpha helix on the surface. So I think you will just have a very crowded absorption, and where you will have eventually alpha helix segments overlapping with each other. I think this is something that it cannot happen. I mean, even, I mean, we've been looking at the distribution of amyloid fibers on the air-water interface. And even there, we have a rigid object on a purely liquid-liquid interface. And we see that we have evidence of isotropic and nematic coexisting, but that this coexistence is not a signature of a first-order transition, is because it's really non-equilibrium process dominate. So I think this scenario of having a nicely aligned alpha helices that derive from 2D excluded volume effect, I think is a very idealized. And I don't understand why are you trying to make that argument? What are you trying to say in that part of the thesis?
It's just more or less commenting on what trying to, because we, with these simple calculations, we see that it's the area that is available is unable to accommodate this many vesicles, alpha helices. So then we thought maybe, okay, maybe this is how it is possible to accommodate.
Yeah.
So,
But the alpha helix, so correct me if I'm wrong. First of all, even alpha helices are flexible object, but if I'm not wrong, there is, I mean, there is a kink or something. I mean, it's not the full one alpha helix, right? There is
it can be broken if that's what, true.
So, I mean, I don't know, in the calculation, did you consider a fully extended alpha helix?
Yes.
Yeah. I think this is, I mean, my personal view on that is this is very unlikely to happen. You will just have a mess at the so, but yeah, I mean, I agree on the calculations, just I think this is very, so another question I wanted to ask is, a bit later in the thesis, you mentioned that fibril formation induced vesicle fusion.
Yes.
I agree. I mean, there is no questions that this happens from the data. I didn't really see, or maybe I was distracted, I couldn't see where. What is the explanation you give? Is this? Did you formulate any possible reasons for that?
No, we still need to discover this, and we plan to do some time-resolved experiments, either contrast matching or cryo-TEM or NMR. But yeah, we are really interested in it. We still are unable to say a lot about it.
What could you guess here? I mean, what would be your guess? What would be the mechanism that fibril can induce vesicle fusion?
Well, if the fibril formation is happening on the surface of vesicles, it's been shown that it can lead to disruption of vesicles, and maybe sometime if they're disrupt, maybe then the fragments meet or form a bigger vesicle, or I'm unsure, to be honest.
But let me just give. I've been thinking. I don't have the answer, of course, myself. I've just been. Do you think it's more like a bridging of fibril or more depletion from fibril which have not absorbed that induce aggregation? I mean, or from another perspective, do you think it comes more from the fibril which have absorbed or from those which have not yet absorbed?
I would guess the ones that are absorbed.
Okay.
But I have no proof.
For sure. But I mean, you think more of a bridging than...
Yeah.
Yeah. Okay.
Yeah.
I see. It could be. It could be many other things, but it's a complicated it's a complicated system. So, okay, good. Now, a point of the thesis where I have a bit of a problem is on paper 3. Okay. All right. First of all, you show in figure 1 a figure. I don't understand why you showed that figure. I would have preferred a lot, by far, figure S3 in the supporting info.
Yeah, well, first of all, this figure has already been published in our group.
Yeah.
So, yeah.
Which
So
one?
it's not S3. So we just use it for this mass preeminent length calculations because it's the highest pH we have.
Yeah.
And it's also low. It goes to not super sufficiently, but sufficient enough low Q value. So
that's exactly my problem.
Yes.
So I wonder, I mean, I needed to understand what's going on here, because if I look at a figure S3, okay, let me check which pH is this one.
It's 7.45, sorry.
So, I mean, here I see that you are capable to go to a Q which is almost, say, 0.00. Yeah, I mean, almost 10 to the minus 3 Armstrong inverse, almost, okay? And then in the figure S1, figure 1, you have lost 1 decade. And that's a pity, because this is exactly the decade where, the range where you will see the Q to the minus 1.
Yes, I agree. But then also we're only discussing this cross-section part of the cylinder.
But I also have a problem. I also have a problem on the cross sections. But first of all, do you? This is X-ray, right?
Yes.
So, what if you have access to 10 to the minus 3? Why don't you put the 10 to the minus 3 or close to that?
Now it's been when I made this. I'm not sure if this sample was measured at all these configurations. I can't honestly now remember why. If there is no, I actually think it was only measured at this configure wide angle and one small angle configuration. We didn't go to very low Q.
Okay, but you know, Maria, you need to forgive me, but my role here today is to make your life a bit difficult. I mean, otherwise, if you get the PhD so easy, there is no fun. So I have to insist on this point, you know, and put the finger into it. Okay, I might understand that the q to the -1, but I also have a slight problem on the larger part of the curve, on the high Q region of the curve. Okay, so can I share the screen again? Let's see whether.
Yes, I think so.
Okay.
I don't know how it's gonna happen, but let's see.
Yeah, I can. So, all right, let me put back this. Do you see my screen?
Yes.
Okay. Now, you have used this model as well in the supporting info, and there is a figure, to your credit, is figure S4, where you attempt to fit with this curve, and at least you fit this part here, the cut off.
Yes. Yeah.
And I see a nice fit. Okay, so the first question I have is, it's not clear from the caption, is figure S3 and figure S4 referring to the same sample?
Yes. Yes.
Okay, then you need to explain me how is this possible, because in figure S3, yeah, we increasing now. People cannot that follow us cannot maybe
They
follow.
should have book. It's page 139 for those who have book.
Yeah. So you know, I mean, in the high Q, okay. So we're looking at amyloid fibrils. In this slide, I'm showing is beta-lactoglobulin. In the thing you're showing is alpha-synuclein. But let's say here we're talking about the cross section. Down to the q to the -1, roughly, we're talking about the same object. And I see in the curve I'm showing you here, there is a cut off which corresponds to this part here. This cut off is the cross sections, okay? And I do not see in a log-log scale, I see that instead of having an increase in the slope, you have a decrease in the slope in your figure S3. And this effect is recurrent even in figure 1. So you have, you don't have this increased cut off in the large Q. You have a smoothing of the curve. And I need to understand what is the reason for that.
Yes, I think I see your point. You're talking about this 10 to -2 crossover that we see.
10 to minus, I'm talking about, I mean, if in figure 1, you see that between 0.1 and 1 Angstrom verse,
yeah,
the curve goes towards a flat thing.
Yes.
Okay.
Okay.
And
That's
instead, the infinitely rigid cylinder. This is the Q -1 part, this is the cross section, will predict that you need to have a cutoff, an exponential cutoff. So I don't see that in your curve.
Yeah, but I think it's now we have nanometers in your.
No, don't worry about this.
Yeah, but I'm saying because what you are showing here, I think that is more at the low Q of the data in figure 1. And that's where we expect Q to -1 if we went high enough. But this, what we see here as a flat part is more some interfibrillar scattering, some contribution from.
Okay, so in your figure, you mean?
Yes.
But do you have a structure factor?
No, we don't think so.
Yeah. So okay. It's surprising, but maybe let me see. What you're saying is that because you know I see in figure S3 that you are able to fit with this. You are able, and now I am a bit lost with the thing because you have QI, but it's a Q to the 0.01. So actually, if I look at the Q region, all right? Okay, I see what you mean. So the cutoff in your part is below, is at Q lower than 0.1 Armstrong inverse, is what you're trying to say, right?
Yeah, I think so.
So, I mean, let's say, if in my curve, we will
Yes.
measure at higher Q, I will
Yes.
also see a
Flattening.
smoothening, okay.
Yes, I
All
think
right, fine.
that's what I'm saying for sure.
Okay. We agree on the experiment. Okay. Now, tell me what is the physical meaning, because as you see, this is not in this model. So this change in slope has to be related to the inside the fibril.
Yeah,
Okay.
but So can you please repeat the question? I'm not sure what you're asking.
So, I mean, we agree now that if I will have measured in a longer Q, higher Q here, I will see the same effect you see in figure 1, figure S1, and figure S3, figure 1, S3, and S4. Okay, we agree on that. So now this smoothening has to come from something which is not in the infinitely infinitely rigid cylinder.
No, yeah.
It has to be inside the cylinder.
Yes,
Okay.
yes.
So, and I see that you fit this your form factor with the Pedersen model, isotropically average cylinders.
Yes.
Yeah. So, and you use a Bessel function, right?
Yes. Yes.
Can you tell me more on what can you say from the fitting between the region 0.1 to 1 Armstrong inverse? What does this part of the curve tell you as information?
In the model, it is mostly affected when changing background. It kind of is, but it's not background in the sense of solvent scattering, because that's been subtracted.
Because you have removed the background,
yes,
yeah.
Yes. But it's more some maybe.
No, but please, Maria, because I really need to understand that. I mean, it's something which has been giving me headache. So you say it might be related to the background, but you have removed the background.
Yes. I mean, I think it's some interfibril arrangement or some, inhomogeneities.
Intrafibr.
Yes.
Intra.
Yeah.
Let me think. 0.1. We're talking about 10 times 6. Yeah, I mean, of course, in that region you will have also the 6 Armstrong 60. Yes, in that region you will have the signature of the amyloid. So it will be sensitive to cross sections, and there might be differences between this type of amyloid I've been reporting and yours. Okay. It's just that, you know, in equation 2, the form factor, I think that this behavior might be hidden in the parameter you have used for the Bessel function. So I like to use model which are as simple as possible, because they're easier to interpret, and it's not straightforward to understand what enables you to capture that part. But to cut the long story short, okay, you see in that region signature of amyloid, because this is the main point. We have still a reflection at, you see at, 1.3 Armstrong inverse is the 4 Armstrong, 4 something. Okay, I agree. But to be provocative, in order to prove that these are amyloid, you need to show also another characteristic length scale. Do you know which one I'm talking about?
Guessing diameter or?
No. I mean, if you show me that there is a reflection at the 4.5 amps, I can tell you, yeah, this can equally be silk.
Okay.
But of course, we know it's not silk, it's amyloid. What are the other lengths that you need to look at to prove that?
Are you talking about the other peak at slightly lower Q-value, which
Yeah.
would be some, yeah, around, oh, I don't, it's like some 10 angstroms.
Yeah, yes,
So that would
yes,
be, yeah, but
yes.
yes, I know we were not able to see it, but I'm guessing maybe we would have needed a slightly higher concentration or maybe a bit longer measurements because this,
Yeah. I mean, I just ask that
Yeah.
peak stuff is tough to get, is much less intense than the peak for Armstrong. These peaks correspond to the parallel beta sheets, not the beta strands.
Yes, yes.
But still, I was curious to know if you at least tried.
Yeah, we did.
So
we
it
were
doesn't show
looking
up.
for them. No, we didn't see it.
Okay. Yeah, I mean, I would have been, and then also, I can say 280 micromolar means, help me out. How much
It's
is
quite high. It's around 4 milligrams per milliliter.
4 milligrams per
I
milliliter.
think.
So it's
Yeah.
0.4, 0.5%. That's not a lot. You will not see that peak. It's really difficult. Okay. Let me see whether I have for sure other questions. Yes. All right. So one chapter that I really like is the one that you have already published. Okay. On the freely joined chain, trying to interpret the behavior of the clusters, right?
Yeah.
So it's
the paper
4. Yes. Okay.
Yes.
That's a very nice work, and also is a very nice effort. Can you tell me a bit more about how you construct the model? So if I have understood correctly, you do this kind of job. You have a rod, okay? You attach at the random point in the length of the rod another rod, okay? Then on rod number 2, you attach in a random point of a third rod, and so on. So it's like a kind of a freely joined chain where there is not topological restriction on where one rod to another one. Okay, is very nice. First of all, you, of course, the rods are not freely penetrating. They have an excluded volume, right?
We consider them to be infinitely thin. We don't exclude any possible overlap. I mean, obviously they overlap but yeah, the monomers can have any coordinate, because basically what we do, we give random coordinates. So we first choose a random coordinate, then we choose a random orientation, and we start building monomers on that orientation. Then we do the same for next fibril. So then we might have fibril 50 and 150. I mean, it's small chance that they will overlap, but there might be some still overlap.
So let me just understand. So that means that if you arrive to a rod number J, okay, this rod number J is attached at the point that is randomly given. And still he has 4 pi solid angle that he can explore, correct? Okay, so there is no excluded volume from the other rods. Okay, good. And this is what the impression I had, and I wonder whether this, let me think now, I don't want to say something stupid. So if there is no excluded volume, yeah, I mean, of course, there is no potential between the rods, right? It's just geometrically. Mhm. I mean, it's quite remarkable that you get a fractal exponent of 2.3, okay, compared to a real fractal exponent that you say is 2.6.
Yeah, average. Yeah.
So given the, I love this part of the thesis, but the model is relatively simplistic compared to reality. Still, it gives you a fractal exponent, which is very close. And I would have expected even, I need to think, 2.3 versus 2.6. So your model is more open than reality.
Yes.
And you have no excluded volume. That's surprising. So more than I totally trust the 2.3. What is really surprising is that you get in reality a 2.6 fractal exponent. So this means that it's almost 3. So this, the fibers are almost totally packed.
Yeah.
How do you explain that? Is the pH or is?
Yeah, well, one for sure contribution is pH, because we know that they are not as densely packed and they also form gel at higher pH. So that is and then we say that they are not that they are colloidally unstable because they're not sufficiently charged. And then we believe that the reason they bundle together in these dense clusters is because there are some hydrophobic patches on the fibril surface, which we don't, there are, that's known. But we believe, so this crowding is hydrophobically driven.
Yes, I also believe so. They must be there because they are so densely packed that even a slightly charged object will. Then you compare it with the Bocage model.
Yes.
So do you, if I'm not wrong, the Bocage model is capable to reproduce 2.6, right? Or not?
Well, we didn't fit Bocage model to the experimental data because we don't really have any linear regime there. So we just did Paulo.
Okay.
But I mean, we get this 2.3 from the modeling from Bocage model.
Okay. Okay. And the 2.3 is from the Bocage.
Yes.
model.
And this 2.6 is just plain power law fit, but to the experimental data.
Yes, it's the experimental. But the 2.3 is the Bocage on your simulated form factor, right?
Yes.
That's okay. Now I got it. I think it's actually is remarkable, and you can get access, of course, to the Guinea regime. You say now, I think 300 nanometer RG. It's figure 3.
Yes, yes, it is. Yes, yeah, yeah.
What about what do the experiment tell you? I mean, did you try to do DLS or TM on this class?
I never done TM myself, but there have been some TMs in group, and that's where people that also very aligned fibrils are also not always, but they are present. But I don't know if DLS would tell us a lot because these clusters are really huge, and how I don't know. But TM was done.
Of course. AEM, did you
no, AFM I never done, and I think it could be useful, especially for the maybe paper 3, where we could also see if there's any influence of lipids on this pitch length or something. But no, I have never done AFM.
Okay. Good. All right. I have a couple of more questions, if I am allowed. The first one is page 168. So I just want to understand here. So if I look at the figure 2, you're saying no steering. So here you were looking at the supernatant, right?
Yes. Yes.
Okay, so no steering. You have a nice fit with the by function. So that means that there is still native alpha-synuclein in the supernatant.
Well, yes, but not depends what we take as native, because we have a little bit higher concentration here than we would expect, but we think it might be due to some dimers or trimers or some small oligomers.
Okay, yeah, let's say still there is protein
Yes.
which is barely starting to aggregate.
Yes.
Okay, and then if you say steering, you
Yes.
just have noise. Is this correct?
No, you put a small magnetic bar into the
Yeah.
Eppendorf tube and you put it above the magnetic plate and then it spins.
Yes, but in the signal, I see there is only random signal.
Noise. Sorry. Yes. So that basically that means that the supernatant is empty.
Okay.
Yeah.
So this means that all the monomers have been exhausted into fibrils.
Precisely.
Okay, got it. Just wanted to make sure that I understand correctly. So that was my impression. So maybe I have one last question for the time being. Is about the last chapter, where you look at the sedimentation of alpha-synuclein with different type of lipid, different mixing of lipid. And I was extremely surprised to see that if you use glycolipid GM1 and GM3, you see a lot of lipid in the sediment, okay? Can you tell me a bit more about these lipids? What is so special with these lipids?
Well, they are, I mean, they are present very abundant in neurons, and it's, I mean, they have very huge sugar groups or head groups, not huge, but some of them. So they have huge head groups, and they've been investigated quite a bit with Alpha-Synuclein. I'm guessing due to their presence in the neurons. So they're involved in signaling somehow.
Yeah. Okay, but why do you?
Why do I think
special in the sense? What make them special to precipitate so highly with the alpha-synuclein?
Well, I mean, there might be possibly some specific interactions. I am unsure what would they be, but I mean, if we are also, when we co-sediment, we are unable still to say if there are any, although we do believe there are because of the other studies performed, but might be due to also their bulky head group, that they might be more easily trapped in the sediment in clusters, or also they might also form micelles together. There is also coexistence of micelles and vesicles when there is a GM present. So there might be also some different interaction with alpha-synuclein and these micelles. So
okay,
could be.
But you confirm me that these lipids are neutral, right?
No, they're charged.
Positively or negatively?
Negatively.
Or deuterionic?
They should be negatively charged, I think.
Okay. Because, yeah, I mean, I thought that since they are glycol, I mean, yeah, maybe we can have it. Maybe you have it reported in the thesis. I don't know.
It might be on page 16. Yes, they are negatively charged.
16, no?
Yes, we have this illustration where
DGM1, oof, that's very small.
yeah, it is, sorry.
So
But
the red is for
negative charge.
negative. Okay, and they only have a negative charge in between the sugars. Yeah, interesting. So, okay, so basically, yeah, it's like in the end, the mechanism could be similar for having an excess of POPS or DMPS. Electrostatic could play a role. Yeah. Okay, good. I think I am happy, and I can pass the words to the other examiner for the time being. Thank you, Maria.
Thank you.
Thank you very much for a very interesting discussion. I will have a podium. Sorry, it was very nice to so like this. Okay.
Could get
So,
some. Yeah.
thank you very much for a very nice discussion. We enjoyed it very much here also in the audience. And now it's the time for the examination committee to ask some questions. And since we have also one of the committee members here in the lecture hall, Susanna, we think it's a nice dynamics to please welcome you to the floor. Meanwhile, we can say a big hand to Raffaele for the nice discussion.
Thank you.
Yes, just a moment. I think I will. Should I?
Yeah, maybe.
Okay, I hope that everyone can hear me also online, hopefully. So I would like to start by thanking you for writing a nice thesis.
Thank you.
As you know, I'm a protein scientist
I do.
rather than a physical chemist. So maybe not so surprisingly, it will be more protein related, but I hope that's okay. It is after all proteins that we study, but maybe there will be something that is, I hope that it will still be in your home turf. So I think amyloids are an intriguing type of protein, or an amyloid formation is an intriguing property that some proteins have. So can you comment on, is there any similarity in sequence or otherwise between proteins that are known to form amyloids?
To best of my knowledge, I think most of the protein can form amyloids under certain conditions. So then I guess no.
So there's no specific type of amino acids that are most commonly found, or?
Well, I'm guessing there must be should be a lot of hydrophobic amino acids, and probably more concentrated at one part of the sequence, as it's believed to be hydrophobically driven self-assembly. But I really don't know if there are any specific amino acids that that bear similar. I'm unaware of that.
Okay.
Yeah.
So with that sense, as you probably know, protein scientists have now a new favorite toy. Can you guess?
Is it
yes.
I would say it's a crystal, but that's not new.
No.
Is it cryotium?
No, it's AlphaFold.
Oh.
Which is the prediction, the ability to predict protein structure from only the sequence.
I've heard of
DeepMind by Google.
Yes, I do know.
Yeah. So then, if there is no, that was my follow-up question, whether you could predict amyloid formation from the sequence. But if you don't think this particular sequence,
I don't think there is.
No. Okay, no worries. I have no idea. So don't worry. I'm not an amyloid person either. Or maybe a protein person, but not an amyloid person. So I wanted to discuss this alpha helix a little bit. So from your thesis, it wasn't very clear to me exactly what it would look like. You have this, I think, on page 10, the sequence.
Yes.
And you have these boxes that show like which part are alpha helical.
Yes.
And then which sequences are important to form the alpha helix. And then I wasn't clear whether that was like multiple alpha helices or one long. It became a little bit more clear in the presentation where we saw the picture.
Yeah, I mean, there can also be different kind of, there can be, it doesn't have to be all. So there's possibility that first 100 amino acids can form alpha helix, but it doesn't have to be all of them involved in alpha helix.
No. So it's flexible.
Yeah, so it's not, doesn't have to be all of them in the same one.
Okay, but you think it can form just one long alpha helix? Definitely. But that is not the part that forms the amyloid.
Well, some of it is, but yeah.
Can it be in an amyloid and still form the alpha helix?
Well, there is. when in amyloid there is this part of first 36 and last 43 are not structured. So they are, I'm guessing that the first 36 might still form alpha helix because they're not involved in beta sheet.
No.
So,
So they
theoretically, I guess, yes.
So what does it look? You have like the beta sheet stacking, and then the other bits are just like sticking out to the side.
Yes.
So which is not completely clear from your thesis, because you discuss it, whether the fibrils actually bind to the membranes. Would that be, you think that would be via the alpha helical portion forming? Or?
Well, it can, I mean, there's also, that's a big open question, but because they can also be just absorption of, maybe they can be electrostatic interaction just with the bilayer and the fibril itself. But it's not excluded that it might still, the sticking part of N-terminal might still form a alpha helix on the lipid membrane. But that's definitely interesting to, I think it might be easy to, not easy, but could be answered with CD, yes, possible, or some other,
And when the alpha helix binds, does it only bind to like the head groups, or does it like go into the bilayer? Because it says in one place in your thesis that it goes in 10 angstrom or something.
There's also some studies that say it only goes to head group layer. Then there's some that it says that it penetrates, but I believe that it does penetrate, because as I said before, there is, when it forms alpha helix, there's this hydrophobic part on one side, and it's quite possible that that side part penetrates in the hydrocarbon region.
That would be a prerequisite, sort of, that is an amphipathic helix, which it is then. Good. Okay. So I was also, as Rafael, a bit interested in this shape change from the discs to the, however you pronounce it, parallel.
Different.
you eloquently avoided mentioning it, so we can stop that to the other shape.
Yes.
Is it, so you have the round and then you get the elongated.
Yes.
Is it the same round that become elongated, or do they fuse?
Yeah, we think it's the same.
And does that add up mathematically?
Well,
if you can
we
measure,
managed
right?
to model the scattering data with this, keeping the area constant.
Okay.
Yeah.
So that makes sense.
Yeah.
So there's no, because when you had the vesicles, then they fused in fibril, when you had fibrils.
Yes.
So it's not completed.
No.
Okay. Okay, good. Thank you. That was easy. Oh yes, and that was just, so I think I will just limit myself to, no, I have 2 more questions.
Okay.
Yeah, so going back to this binding to the discs and also to the vesicles, you have this all or nothing theory.
Yes.
How does that, could it be that when you have the disc to the other shape transition, could it be that we only see the shape transition when you have a certain number of monomers bound?
Yeah.
Or what are you following? I don't remember completely. Are you following the shape change or the binding?
Shape change.
So could it be that there's only, the shape change only happens when you have all bound?
Well, I think it could be, but I don't, because we also have different length of these shapes. I think they're also, not all of them accommodate the same amount of proteins.
But the discs are not the same size either.
That's true. That is true. I think it would, maybe it wouldn't be as elongated. Maybe it would, because in some cryo-TEM images we see,
yeah, please.
Something that looks like this, kind of, and then we probably have some absorbed here and none here, and that we see at some later points.
Okay,
Not
so
at
then
the
it has started
time.
changing shape before. Okay. And when it comes to the binding to the vesicles,
Yes.
but then it was fibrils, right? or it was both?
Both we
Yeah.
studied, yeah.
Is it
But we
all or
didn't-
nothing there as well?
We don't know.
You don't know.
No, we don't, because we just look at the end of fibril formation process,
Mm.
where we see some already fused, and we are unable to say much more about it.
Do you have any idea how this would? You say it's cooperative. Do you have any speculation of how sort of
Well,
what happens?
why it's cooperative?
Yeah, how it helps the mechanism behind.
Well, I think this shape is not very stable to start with, but I also know in our group it's been shown that the adsorption is also cooperative. So, so I'm guessing also where it doesn't look stable. I think it's more about this line tension energy, and the other just don't think it's the most stable. To have it half.
Yeah. Okay. And then just a final question. I had already had thought about this, but you also mentioned it about doing time resolved studies.
Yeah.
How would you do that?
Well, we have.
You have some sort of time resolved, but on very long time scales, like with hours.
Yes,
on
because this where we see vesicle fusion, that is where we have very much slower aggregation than when. So the time resolved, at least neutron at these conditions would be, unless we have samples prepared at different time points so that we measure them. But then I'm also thinking we could try to see with some cheaper methods at neutrons if that would happen also at this lower pH where we have faster aggregation. So if we see at the end that vesicle fuse, then maybe we could try neutrons there. Otherwise, taking snapshot of with cryo-TM at different time points. And also following diffusion with NMR is also, because then if we have bigger vesicles, it will be slower. And
how fast is the when you need to start adding the seeds to start getting, so how fast is then the fibrillation process?
I haven't performed any of THC that would maybe tell us that. We just had them incubated for 2 weeks and then we measured them.
I was sort of thinking whether you could do some time resolved sacs or something, or sounds that where you have like a rapid mixing.
Yeah.
So you rapidly, just fluid handling, basically. You rapidly add the seeds and then follow what happens.
Yeah, exactly. But that's I think
in
first
real life
step would be maybe to do THC fluorescence to see exactly if the time lag time is too long. Maybe then do it. We definitely need to do some lots of studies to see, like, to choose right conditions as well, and so can't measure for 2 weeks.
Yeah. Okay, I think I will finish there.
Thank you.
Thank you for a nice discussion.
Thank you.
Thank you, Susanna. Very nice. Yes, so please, now I would like to ask Vito to ask questions. And
Yes.
I will soon pin you here, so you are just a moment. It's difficult with the...
Can you hear me?
Yes.
Perfect.
Now, please.
Okay.
Welcome.
Yeah, thanks a lot, and thanks a lot for the so far nice afternoon, Maria. So it was very nice to listen to your work and also to read through your thesis. And I actually now have 2 copies. I got also the first one that you have been sending out, so that's very important. So I mean, since we was one of the first words mentioned by the others, I'm also an amyloid person. I'm also an X-ray person. So it was a very a great pleasure to read the thesis. So, but I'm also a guy that likes a lot kinetics of amyloids. So I have a couple of questions related to that. But I would like to start with mentioning a paragraph that we have in page 15. And I was in this paragraph 2.3 where you say fibril formation, and then you have this subtitle that is fibrils were stirred, not shaken. And then I was expecting a bit of an elaboration on why you were
Ah.
using steering and not shaking. So can you elaborate on this?
Well, I don't know if I have a good reason for that. I think I was just following the protocols of that people have done before me, and yeah.
Can you, I mean, can you guess a little bit what can be the main difference between the two approaches? Because I guess when you say shaking, you are thinking about Eppendorf shaker or
Yes.
shaking of the plate reader, I guess.
Well, if in play both,
Yeah.
but I'm usually having them in Eppendorf tubes. I would guess that there's steering would induce more fragmentation, which would further increase the kinetics rate even more.
And that's a very nice question because it's connected to a question I have at page 51.
Yeah.
Here it's very nice. So actually, I like the answer you gave me. But then you write here this: when steering is introduced during fiber formation, aggregation kinetics is significantly faster compared to the one occurring under quiescent condition. And then you say, we explain this observation by loss of a catalytic surface for the new fibrils to form.
Yes.
So my point is, that's true. You lose catalytic surface, but you introduce steering that you just say that is also inducing fragmentation.
Yeah. But in the case when we have steering, we have the fragmentation, and then we don't have sedimentation. So we have even two conditions that would increase the kinetic rates.
And that's good. And do you have any guess which one is the dominant one?
Well, at this condition, the secondary nucleation in general is more dominant at quiescent condition than fragmentation. But that is just without any quiescent condition. So I don't know.
Yeah, that's fine. It was just to see if you had a little bit of a picture of how this could have, I mean, what kind of effect you can have on the kinetics. Okay, good. And yeah, let's go back to page 34, actually. And as Raffaele already mentioned, I really like the CD data. They are very nice. But I would like you to comment a little bit on the linear combination that you have in panel C, because the linear combination is the blue data, right?
Yes.
And the linear combination is ending around 200 nanometer.
Yes, and there is a reason for that. And that's all these, but in figure A, I think you also see that the higher lipid protein ratios are ending at lower wavelength, and that's because we have at this low wavelengths, we have also lipids are somehow influencing the CD signal, so just noise.
So you're saying that you have a lot of scattering, and then you cannot follow below that. Okay.
So it's just the noise, so it just looks not pretty. That's why we just.
Okay, but by the way, this is a very, actually you can sell it a sort of quantitative way to look at the effect of the charge on and actually I have a question on panel B. So do you have any, let's see, do you have any, because I think this one is as a function of the charge. So you change the charge in panel B between the red and the, right?
Yes.
So do you, to see if this trend is real, do you have other measurements that maybe can show that a specific trend with charge?
You mean other techniques or?
No, I mean another set of data with a different.
I mean there's there's more data points for the 30% charge. There are more data sets because that's the one I investigated mostly. But for this one, no.
Okay. Yeah, that's completely fine. Yes, and then a question related to the kinetics, and it's page 45, because I was, to be honest, I was not shocked, but I was surprised about this, the fact that you basically have no effect on the packing of fibers due to the lipids. But I cannot, I mean, it's difficult for me to see no difference in the kinetics, in the aggregation kinetics, because I think if you have an object there, even if you do not have a direct interaction, you change a little bit the bulk property of the solution. And I would expect that the time scale of the process can be quite different between with and without lipids. So do you have any thoughts about how?
Well, it's why we actually use seeds at this condition is because lipids are not really catalyzing the aggregation at this higher pH. So I haven't checked it myself, which is something I should probably do. But so for that reason, I'm guessing it wouldn't affect the kinetic significantly.
Yeah, I think I will do the experiment, because if they affect the kinetics, it's another PhD project probably, because it's extremely interesting to see that you basically do not change the microscopic structure, still having this object. That, as I said, even if you do not have direct interaction, you always. I mean, I expect a bulk effect that should change a little bit the kinetics. Yes, and talking always about this, do you have any, because if you do not see any clear effect on the structure, often you have a different stability of the fibrils. So if you form in different configurations, so do you have any idea if in one case you have more stable fibril than the other?
I don't.
Yeah, that's this is a couple of suggestions for new projects.
They were very good.
Maybe I'm talking mostly to your supervisor as well, but that's good. And yes, I have a couple of considerations left on. I like a lot the last work, the work on the cluster, but I would like to know a little bit more. Because, so correct me if I'm wrong. So you form the fibrils at pH 5.5, and then you still look at the clustering at pH 5.5.
Yes.
Okay.
Everything
so,
is 5.5.
Okay. Yes. And okay. Because there is all this new. There are a number of papers now looking at maturation of fibrils and maturation of the aggregates during the even in vitro experiment. So I was actually wondering if I don't think you have, but do you know if there is any difference in the secondary structure of those clusters over time?
I mean, I wouldn't guess so, because I believe that they are very stable amyloid fibrils, but I haven't checked it.
Yeah, I mean, 2 years ago we published on Alpha-Synuclein at pH 7.4. Actually, if you look at the plateau of the kinetics, if you do a structural investigation, you actually see change in the secondary structure. So they actually develop even if the kinetics reach the plateau. So my guess is that this cluster formation is actually, it's not only a morphological thing that you see, but it's actually something more, let's say, more deep in terms of structural change. So I think it would be nice to see the FTIR spectra on this kind of aggregates. And then a more general question. So is pH 5.5 something relevant if we look in vivo setup?
Yeah, there are some places like endosomes and lysosomes. No, endosomes mostly. They have 5.5, late endosomes.
But would in vivo some aggregates experience pH jumps or like a stable pH around? I mean, because having a constant pH, I guess it's also in an in vivo setup, it's quite difficult. I mean, it depends. I mean, the pH, it's locally changing quite rapidly. So another suggestion can actually be working with pH jumps around these aggregates and see how it works. Yes, and I think I'm satisfied. And I also thank Raffaele that has been stealing some of my questions, and also Susanne before me, but that's perfectly fine. And congratulations again for the work.
Thank you. Thank you.
Thank you very much for a nice discussion. So now I would like to welcome Anela to ask questions.
Thank you. Can you hear me okay?
Yes, I will
Brilliant.
pin you.
I'll also start by congratulating you on what I think is a really elegant and fantastically interesting piece of work. I've really, really enjoyed reading it. So well done. And I'd also like to thank the other examiners for stealing almost all of my questions. So by going last, I've basically, I think I've got the easy job because I haven't got that much left to ask. So just to give you a quick idea of where I'm coming from in terms of what I'm interested in, I'm an X-ray neutron scatterer and do a lot of work on lipids, but also on soft matter solution scattering as well. And I've got a few questions around the lipids that you've used, and some questions around things that you may not have done, but you may want to consider, or maybe you have an idea of how they might work. So the first thing I wanted to ask was, in your vesicles that you made, you used a mixture of POPC and POPS. I completely understand the rationalization for having to use a charged lipid. I was wondering why the selection of a lipid with an asymmetric tail. Was that important, or was that just something that correlated with what's been done in the literature?
Yeah, I mean, so there are some also practical reasons for example, using the MPS, which is the fact that they can be deuterated, and it's something we want for contrast matching. But I mean, it's also, I think, good to do some systematic studies on different lipid properties as well, to learn about some possible selectivities. But it's also in the literature, there's people are using various lipids.
So you mentioned the DMPS. Obviously, the phase transition temperature of DMPS is quite high.
Yes.
So was everything that you did with the disc micelles done above the phase transition temperature of both lipids?
Actually the preparation was, and the kinetics neutron experiments were as well. Yeah. So yeah.
Okay. Right, that's absolutely fine. Do you think, obviously, you know, there has been work done on these systems in the literature before, just going back to the idea of the fact that you have an asymmetric tail. Obviously the binding you would expect to be to the head grip region, but do you think the tail might have any effect on how the, say, the vesicles behave if you have, for example, something like DOPC or a symmetric tail?
Well, first of all, in all the systems, their tails are the same. It's just if I have vesicles, all the lipids are the same tail length. It's just, well, we see, for example, that this difference between PO and DM resulted in the completely different membranes, such as we have disc micelles in short, and yeah. But what it can influence the bilayer thickness, of course. And also this saturated, unsaturated can also influence the packing of the lipids in the bilayer, meaning more or less expose the hydrophobic region.
Okay, brilliant, thank you. So looking again at the, luckily all the questions I had about the vesicle fusion, which you've now been asked on a couple of occasions, I'll take your answer as read now that you have got some experiments that you've got planned to do. But I was interested to see that you prepared your vesicles for that experiment using sonication. Yeah. And looking at the graph on page 99, where you've done the very nice DLS data, you have obviously this slight increase in the hydrodynamic radius when you add in the Alpha-Synuclein. I was really interested to see how monodispersed they appeared despite being prepared by sonication. Maybe. Yeah, because I would have expected them to have been more polydispersed. Did you do multiple measurements and put some, could you?
Oh, I usually check DLS whenever I prepare vesicle just to be sure. Oh, now I don't have a number, but they are a bit more polydispersed than when they are extruded, but not that much. And I don't have the number in my head.
That's what it's just maybe in my hands, every time I've prepared anything by sonication, it's always been relatively polydispersed compared to extrusion. So that led me to my next sort of query around this particular experiment. Is you've got quite small vesicles here, so that we're looking at, you know, a diameter of just over 50 nanometers.
Yes,
so this has got a very high geometric curvature, and things with high geometric curvature are more likely to fuse when they encounter each other. I wondered if you tried with larger vesicles, maybe extruded vesicles, where you actually have almost a kind of a lower curvature, so the protein's seeing a flatter interface.
There were some experiments done with the larger vesicles, not still small, but larger than this. But we were not looking at vesicle fusion in these experiments. Yeah, when we also considered this possibility that just the small unstable ones would fuse rather than big ones. But we haven't investigated that yet with bigger vesicles.
It might be a nice thing if you're going to do, for example, some
definitely.
rapid mixing of vesicle and protein and look at fusion. If you change the geometric curvature, it might give you a handle on the fusion mechanism itself. Okay, so moving then to your last piece of work, which I thought was really, really interesting around the co-sedimentation of the lipids. I appreciate that obviously there was no kind of logical pattern to what co-sedimented with what and how reproducible it was. I was wondering if you had to kind of hazard a guess at why that was and how you might improve that for the future. What would you do?
Well, I'm thinking that also we don't really have, first of all, control of the number of fibrils that are formed. So in the future, I'm pretty sure I will just stir the fibrils to make sure that they are as much more or less as can be. So that might influence also sedimentation. Yeah, I don't. What was the question?
I was just really interested that you know, obviously the POPC POPS mixture, you know, you're getting quite a high fraction.
Sometimes, yes. Sometimes
sediment, yeah, sometimes that, and then sometimes, and so there doesn't seem to be one lipid that's driving it.
No, yeah. So and it's especially in the figure 5A, it seems to be more random, where we don't stir. So as I'm saying, but not when we have gangliosides. So maybe there is still some really selectivity with gangliosides that is does not really need all the fibrils to be formed.
Yeah.
So, well.
Okay, and then my final question was just a very general question around. I think your X-ray neutron scattering data is beautiful, and I think you've done a great job in fitting it. But quite a few times throughout the thesis, you used the phrase, you know, that the model was a good fit for the data, which I agree with. What were your criteria for deciding whether something was a good fit?
Yes, well. But if I would do it in SAS-view, I would consider chi-square. But sometimes we would model in MATLAB only. In most of the cases, we're just simulating. But in SAS-view, you can still get some approximate estimation of the I mean, obviously, if the parameters are reasonable, but yeah, basically how it looks. Yeah.
Okay. Like I said, most of my other questions have already been asked, so that kind of leaves me with these ones. So that's me done. Well done again. Thank you
so much. Thank you.
Thank you very much, Anela. Really interesting questions we have heard, and also very good answers. We have one member that a deputy member, but in the audience, Marija Dubackic. I just would like you to ask you if you would like to ask one question, or you will pass. You have no further questions. Okay. Then I'd like to ask the audience if there are anybody in the audience that would like to ask a question. No, nobody. Okay. So I think then we will end this part, or this official public event. And I would like to thank everybody, and especially Raffaele and Marija, and the committee members, Vito, Anela, and Susanna. So thank you with a big applause. And now this part is ended, and you in the audience are welcome to go to the coffee room at the physical chemistry department.
Shouldn't it be in Cafe Este?
Oh, sorry.
Is it?
I'm not latest update. It's in Cafe Esther. And for the committee members and Raffaele, we will. Sorry, I'm out of focus. Okay, we will now close this Zoom link, and then you have got the second Zoom link. Yeah. So you log in, and we will be here. Susanna and me. We will stay on, and the supervisors. We decided to stay on here. So okay. It's
See you in
difficult
the
to
other
end.
channel.
Please, everybody. Thank you once more. Bye bye. Okay.